Hi, Dr. Wang here. Today we are going to do this assignment called Conclusion Lab 7, analyzing the gel image obtained from restriction digestion of the PCR product. Before attempting this assignment, please complete APE 7. This is going to simulate the actual lab and generate predicted data so that you can use that and compare that to the actual data. So all the sequence files are provided in this folder. So if I click on that, you can see that this is the sequence for the empty vector. This is the sequence for the PMSH2 carrying the wild type MSH2. And we also have 4 different versions of the mutant. From mutant 1 to mutant 4. So you're going to analyze those sequences in APE7. Even though there's no separate training video for APE7, there are training videos associated with APE 4, 5, and 6 that's going to help you with this assignment. In the first question, you're going to identify the type of mutant that's present in mutant 1, 2, 3, and 4. All right, so in question number 2, you're going to first simulate the PCR reaction using APE. So the instruction for this is in, I think, APE5 primer and PCR. So once you have simulated the PCR product, you're going to compare the wild type with the mutant in NEB kata and identify which of the following enzymes will allow you to differentiate the wild type PCR product from the mutant PCR product. All right, so and once you have identified that enzyme, for example, you identified an enzyme for mutant 1, and then you're going to do a simulated digestion on the PCR product with that particular enzyme, and then this is going to give you the answer for question number 3 is how many fragments will be generated through that simulated restriction digestion and what are the size of those fragments. So at this point, please remember to save your simulated gel image because you're going to use it in conclusion for lab 7. So now let's move on for this assignment. So this is a simple matching question asking you for the identity of all 3 samples. By this point, we now have enough data to answer this question. So what is sample A? Is it the empty vector? Is it the wild type or is it the mutant? If you are still not sure, then you probably will choose this one. But this is not the right answer though. At this point, we should know exactly what it is. All right, so and this instruction, data file and instruction for lab 7 is going to help you through the entire analysis so that you can answer the questions in here. So let me go through this quickly. So here are the actual data. So this 4 files representing 4 different mutants, right? So the samples loaded onto each gel is actually given on the comments page. So this is the sequence of how those things are loaded. So the first lane contains 100 base pair DNA ladder. We're going to come back to this in a moment. The second lane is sample A digested, followed by sample B digested. Right, so I'm going to come back to here. So sample A digested, sample B digested, sample C digested, and followed by 3 undigested samples A, B, and C. So they're loaded in exactly the same sequence. So one gel is from mutant 1, the other from mutant 2, mutant 3, and mutant 4. Right? So at this point, we don't know really which one is which, but you should have known from APE 7. Right? So for example, if you have chosen BMR1 for mutant 1, then this gel right here, BMR1 gel, is actually for mutant 1, right? So it depends on your answer from APE7. You can assign mutant identity to each of the 4 gels. So you're only going to pick one for your analysis. You're not going to do all 4. Just pick one of the images as your raw data to use to update your lab notebook for today. So here, this notebook update should contain method for lab 5, 6, and 7. For the results, you're going to analyze the pick a gel image, right? So for example, we're going to pick the first gel image, and then we're going to analyze this gel image. First of all, how many fragments is presented in that lane? So we can see that sample A, right? So if we take this gel as an example, sample A, which is lane 2, there's nothing. So then it will be 0 fragment, and then not apply, right? So, and then for sample B, we can see that there is a single fragment. And for sample C, we can see that there are 2 clear fragments. So this is how many fragments, but how do we estimate their size? For this, we're going to use the ladder. So I'm going to click on this ladder. So this is going to bring us to the manufacturer's website. And then on here, we're supposed to get a document showing the image of this ladder. So this image should be right here, document, right here. All right, so we have the image right here. So we can see that this ladder has 2 bright fragments, which is right here. So the lower one is 500 base pair, the upper one is 1,000 base pair, and in between, this is 100 base pair apart. So the next one is 600, 700, 800, and 900, and so on. Right? So we can come back to this gel image, and we can see those 2 bright fragments right here. So this one is 500, 600, 700, 800, 900, and 1,000. So to estimate the size of this fragment, all you have to do is to sort of come this way. Okay, it's closest to this fragment right here. Therefore, its estimated size is going to be close to this fragment. So for estimation, we don't have to be very accurate. We normally just estimate them to be 100, 200. If it's in between, we might say it's 350. Right? So estimation is not supposed to be very accurate. So this is how to fill in this table. Just pick one gel image and then analyze the data presented on that image. So how do we determine which one is the mutant, right? So we have sample A, B, C, which one is the wild type, which one is the mutant. So for that, you're going to compare that to your result from APE7. So you have your saved simulated gel image, right? So if you picked BMR1, then you're going to use the saved image for BMR1. Right. So here, and then that image is going to tell you how many fragments will be generated by that digestion, and then what is the predicted size. So here it will be very accurate. It will down to the very last digit. It's like 167 base pair, for example. So you can do that fairly easily for PMSH2 and PMSH2 mutant. Right? So just pick whichever mutant that you're working with, depending on which gel you picked. So for bonus point, this is PRS 413. So this is the empty vector. So this is where the question comes in. Do we expect the PCR to generate a product for this empty vector, right? So the hint solving this is, can you find primer location on PRS 413? If you can find it, then PCR should work. If you cannot find those 2 primer locations, then PCR is not going to work. All right, so here's the end for this tutorial.